Right, so it gives me great pleasure to introduce our speaker for the fourth week this term, Jim Al-Khalili, who's becoming a great friend and colleague, but is well known to us all. In fact, he's in danger of becoming a national treasure. If you don't watch out, Jim, you'll be on Desert Island Discs. So, this is the Life Scientific, of course, that we all know and love. And Jim has began life as a nuclear physicist, and then worked on larger and larger length scales, and he's worked up to biology now. And of course, that's still pretty small scales. We're interested in quantum biology in particular, and a mutual project that Jim and I are working on involves that. Jim has written a book, Life on the Edge, which I very much recommend to you. It's an excellent book. But that came out 5 or 6 years ago, and he's got a new book out, which is called The Joy of Science. So, and that's also, I think, going to be a very interesting book. Jim, welcome. It's a great pleasure to have you here. The floor is yours.
Thank you very much, Simon. Well, good afternoon, everyone. It's nice to be in front of such an illustrious audience, and actually the first time Simon and I have met in the flesh, despite being on a project together for half a year now. Yes, so I mentioned this, I've got this new book out. So I've spent the last 6-7 weeks traveling around literary festivals, giving talks about that book to sort of public audiences. So it's actually quite refreshing, quite nice to be able to give a more serious talk today. It's really a talk in 2 parts. I'm going to be giving an outline of the history of quantum biology, what it's about, what it is, what it isn't, how it began, and sort of through rather sort of a bumpy 20th century. And then the second half, I'm going to be talking about what we're doing now in quantum biology, and the sorts of, and show some recent results from papers we've published in the last year or so. When it comes to quantum mechanics, biologists tend to shy away from it. They, I think, but historically, quantum mechanics developed in the 1920s and 30s at the same time that biologists were developing genetics and molecular biology, and they sort of felt they didn't need any help from quantum mechanics. They're doing very, and they've done very well in the last century without quantum mechanics. Physicists have shied away by and large from biology, although there are such things, biophysicists, because biology is messy and complicated, and they much prefer things that they can control in their labs. And chemists who sit in between physics and biology tend to wonder what all the fuss is about. So, so what is this new thing called quantum biology? Well, I mean, historically, molecular biology has tended to be essentially balls and sticks models of atoms, and the sticks are the chemical bonds between them. Of course, you're all probably familiar with the famous Crick-Watson double helix structure, which is obviously more complicated than this. These days, of course, molecular biologists use more sophisticated computational techniques to model their molecules. This is a large biomolecule, maybe 100,000 atoms, different color-coded atoms, and there isn't any quantum mechanics here. Well, there may be something to do with the chemical bonds, but essentially it's stopped running now. But you see there were sort of vibrations. It's a purely sort of still a classical model. So my question, why it is that when we're talking about the scale of individual atoms and molecules, why there isn't more quantum mechanics? Quantum biology, first of all, what it isn't. So if we say that quantum physics is somehow the foundation of reality, by the way, I'm leaving general relativity and gravity and the macro world aside in this talk. So if quantum physics underpins ultimately organic chemistry scaled up in complexity is molecular biology is life. So naively you think, oh, Jim's arguing that life must depend on quantum mechanics because you can see the hierarchy. But of course, inanimate matter also goes down at some point. When you zoom in, you're going to hit the quantum world. This isn't what we mean by quantum biology. Quantum biology is about non-trivial quantum effects playing a functional role inside living cells where really, historically, we wouldn't have expected them. And I'll say something about this later on. Clearly in a hot warm, complex environment like a living cell with thousands of biochemical reactions going on, why would you expect quantum coherence to last for any timescale that might have a functional role in biology? Well, let me go back then and say a little bit about the history of the subject and how it first started. Of course, going all the way back to the beginning of the 20th century, there were two reasons why there was an interest in a potential role that quantum physics might play in biology. The obvious one was the success of quantum mechanics. So, mid-1920s, the physicists, Bohr's protégés, sort of leapt out of their physics labs and offices and thought they'd go and give the biologists a helping hand, because clearly they're going to need some help, because physics had solved, quantum mechanics had solved all the problems in physics and chemistry, and they thought maybe the biologists could do with a hand as well. The less obvious one was this new movement called organicism. There were two schools of thought. We go back to the beginning of the 20th century. There was the idea of reductionism, breaking something down to the sum of it's nothing more than the sum of its parts. On the other hand, you had the fading notion of vitalism, that there was something about life that was special. There was the spark of life that distinguishes it from inanimate matter. But in between those, sort of a halfway house, was this idea of organicism. It says that, yes, there is something different, something that we have yet to understand about why life is special, but it must only depend on maybe hitherto misunderstood or incomplete understanding from physics and chemistry, but it has to rely in some sense on physics and chemistry. This idea of the organicists, I guess, goes back to Bertalanffy, who's an Austrian biologist, one of the founders of what's called general systems biology. And he claimed that biology needed some fresh impetus, some new ideas, some new principle that underpins what life means. There was also this sense of optimism. Biology, as a physicist, we tend to obsess with what happened in the early 20th century in physics, relativity, and then the full-blown quantum mechanics in the 1920s and quantum field theory, 30s and 40s and so on. And we forget just how rapidly biology was also developing at that time, molecular biology. So clearly there were experimental advances in biology, and particularly overlapping in biophysics as well, that suggested we need to work harder at understanding biological systems down at the molecular scale. I'm going to go through a timeline of some of the main landmarks, key dates in the history of quantum biology. Essentially, we can start, as one always must, with Niels Bohr. Niels Bohr inspired a number of his students who came to work with him, in particular people like Max Delbruck, who of course then went and worked in biology, but also Pascal Jordan. Bohr gave a talk at a conference, which I pull out this quote. He says, "Before I conclude, it will be natural at such a joint meeting of natural scientists to touch upon the question as to what light can be thrown upon the problems regarding living organisms by the latest developments of our knowledge of atomic phenomena," by which he means quantum mechanics, which I have here described. So he hints, he gives, as Bohr would do, he'd just give these vague ideas that people would take up and run with and develop. And one of those people was Jordan, who published the very first paper, looking back now on what we might call quantum biology in the early 1930s. Now, Jordan, of course, was made famous because of his association with Max Born and Werner Heisenberg. So these famous papers on matrix mechanics in the mid-1920s, that sealed Jordan's reputation as one of the founders of quantum mechanics. He then went on to publish his paper in quantum biology. He had some idea, some theory that indeterminism could be could somehow be used, or indeterminism, or the ideas of Bohr's ideas of complementarity could be used in biological systems. We might regard Pascal Jordan then as the father of quantum biology. Maybe we should be celebrating the fact that he was the father of quantum biology, if not for the fact that he was a Nazi. And I don't mean a keep your head down, Werner Heisenberg type, I don't want to get involved sort of thing, but nevertheless not speaking out against fascism. No, Jordan was as far as we can tell, fully paid up fascist. And of course, in a sense, quantum biology, if he was the father of it, it sort of suffered. Probably there are parallels with eugenics, I guess. It was seen as it fell out of favor for many decades. But another physicist who showed an interest in biology and whether quantum mechanics might play a role in biology, of course, was Werner Heisenberg. With Erwin Schrödinger. You can see this is a page from a notebook of Schrödinger's. He was preparing for a lecture. He wanted to understand genetics and the structure of chromosomes and so on. He became very interested in biology. He published a few papers on the subject, but of course he's most famous for having written his book, What is Life? And that was not until 1944. And again, there's a very nice quote here from the book. He says, the living organism seems to be a macroscopic system, which is in part of its behavior, it approaches purely mechanical as contrasted to thermodynamical behavior, to which all systems tend, as the temperature approaches the absolute zero and the molecular disorder is removed. Essentially, living organisms, say like a living cell, behaves or has a sort of structure that is quite different from sort of the thermodynamic chaos that you might expect for something at that temperature. And it behaves more like inanimate matter of equivalent complexity near absolute zero, where we know quantum effects kick in, superconductivity to superfluidity and so on. So Schrödinger's idea was that somehow, living organisms were not like steam engines. They're not just taking low entropy energy, putting it to use and giving out waste heat like a steam engine. They were somehow able, and Schrödinger doesn't really, apart from talking about things like aperiodic crystals, he doesn't really go into any detail as to how life might reach down through the randomness of thermodynamics down to the sort of the base quantum level where there might be some organization, some rules, some structure. That was in 1944. Since then, there have been a few key moments in the history of quantum biology, but they've pretty much gone unnoticed. So certainly, I need my glasses there because my slides on my laptop here, the text is so small I can't see it. Certainly, Crick and Watson were very influenced by Schrödinger's book, What is Life? Of course, they didn't suggest that the double helix structure had any issues or needed any explanation in terms of quantum mechanics. But it wasn't until the 1960s when the Swedish, he's a physical chemist, Per Lövdin, published a paper which took Crick and Watson's idea that Crick and Watson had suggested that the hydrogen bonds, these are the rungs of the ladder, the twisted ladder that is the double helix, the two DNA strands, the hydrogen bonds that hold the strands together, essentially for a nuclear physicist like me, they're protons. I don't care about electrons, I leave them to the chemists. So the proton is the glue. It provides the glue between the two strands. Crick and Watson had suggested that proton could transfer from one strand to the other. So the bond between the nucleotides, A, C, T, and G, and the proton tends to sit closer to one side than the other. It could jump across to the other side. And then as the strands split, when they're unzipped by an enzyme called the helicase, that proton could find itself on the wrong site. And that could, Crick and Watson suggested, lead to a mutation. Well, Lövdin suggested that proton could transfer via quantum tunneling. There's an energy barrier, there's a double potential well, and that proton could maybe tunnel through the barrier. But it was very hand wavy. He didn't sort of add any calculations. This is mid-60s, and those calculations didn't take place until recently, which is the subject of the second half of my talk. But another big step forward was by two spectroscopists in America, DeVault and Chance, who first saw experimental evidence of electron tunneling in enzymes. So enzymes are the workhorses of the cell. Right? They catalyze chemical reactions, they speed them up by many orders of magnitude. And one of the tricks that had been suggested was that enzymes make use of quantum mechanics, in particular, moving an electron from one part of a biomolecule to another in the enzyme's process of breaking up and building proteins, that quantum tunneling might play a role. The theory that supported this was developed in the mid-1970s by Hopfield, and round about the same time, you might think this is not related at all, but it was established that certain animals utilize the Earth's magnetic field to help them with their sense of direction. They use the magnetic field of the Earth to navigate. In fact, famous paper in the journal Science by two German ornithologists, the husband and wife, the Wilczkos, carried out wonderful experiments which confirmed that this is what happens. In particular, the European robin, which migrates from Scandinavia down to the Mediterranean every autumn, is able to sense the direction of the Earth's magnetic field and use that for navigational purposes. Even that was deemed to be sort of pretty weird and wacky. How can the Earth's magnetic fields, being so weak, have some influence on chemistry inside a living system? But many experiments after that confirmed that in fact they were right. The question was, where was this chemical compass? The suggestions that it might have been magnetic crystals lining up alongside nerve cells, which could be affected by the orientation of the bird in the magnetic field and stretch the nerve, sending signals to the brain. But in some cases that had been ruled out. That couldn't be the answer. By the late 80s, a team in Berkeley had shown that enzymes not only are able to cause electrons to quantum tunnel, but protons as well, 2,000 times more massive. And so this is the team of Judith Klinman in Berkeley. So they showed that proton tunneling in enzymatic action they confirmed that in the lab. By the year 2000, another group had proposed a model that could explain the chemical compass on in the birds. And of course now the work that is being done in this field is carried out in the chemistry department here at Oxford. Peter Hoare's group, Peter, who's only just been elected a fellow of the Royal Society, which is very well deserved. But his group have suggested that the mechanism that allows these birds, and in fact other animals, insects and marine animals, to sense the Earth's magnetic field is down to a mechanism that's in the bird's eye, in the retina. So it's light activated in a protein called cryptochrome, which is a light sensitive protein. The idea is that a photon of light enters the retina, hits one of a pair of electrons in an atom in this biomolecule, the cryptochrome, knocking it out. So these two electrons are together, so they're sort of entangled, maybe spin up, spin down, knocks one of them off to an adjacent atom within the same molecule. And those electrons remain quantum entangled. And it's the ratio of whether they are in a spin triplet or singlet state that is very sensitive to the orientation of the bird, and hence this cryptochrome, and hence this pair of entangled electrons within the magnetic field. So the external magnetic field changes the balance between the different spin states of this pair of electrons, sending different signals to the bird's brain, telling it which way to go. I mean, it seems, when I say it, it sounds far-fetched. But firstly, it's the only theory we have at the moment in terms of magnetoreception and how that might work. And secondly, it makes sense that the sums work. The difficulty is, how do you confirm this experimentally? How do you measure, quantify entanglement within a protein within a bird that's flying around? You can carry out experiments to check that the bird is indeed sensitive to this magnetic field. That's how a lot of these experiments were done. You'd capture these birds in mid-migration, and they would put them inside these in their laboratories within some setup with Helmholtz coils that have a magnetic field that can be made to sort of cancel out the Earth's magnetic field. And these birds lose all sense of direction. So when they want to fly off, they don't know which direction they're going. Switch off the internal magnetic field, they sense the Earth's magnetic field again, and somehow they have some more coordinated direction in which they want to fly to continue their migratory journey. So that's established. But how do you test for quantum effect like entanglement in the retina of a bird's eye? I mean, it will be great. It's a wonderful story that quantum entanglement, Einstein's spooky action at a distance, helps the European robin find its way south every autumn. But because you want something to be right doesn't make it so. In 2007, a team in Chicago and Berkeley, Greg Engel and Fleming, carried out an experiment in which they showed long-lived quantum coherence in cryptochrome. In photosynthesis. So we know that plants and bacteria carry out photosynthesis. They capture sunlight, turn it into chemical energy. The first step in photosynthesis involves taking that photon of light and delivering it to the reaction center within the cell where it can be used. And the argument is that if that photon would just, like in a pinball machine, if it was to see if it could find its way to the reaction center before being lost as waste heat, then you wouldn't see the high degree of fidelity and efficiency of this stage of photosynthesis. Photosynthesis is a hugely complicated multi-step process, but this is the first step. Chlorophyll capturing the photon and delivering it to the reaction center. And it seems to do that with unerring accuracy. And so what their experiment claimed to show was that in fact that photon, what happens is that it excites an electron in an atom, and that creates what's called an exciton state. And that exciton state is then put into a superposition of multiple pathways. It's like the two-slit experiment, but with multiple routes, sort of Feynman paths, all ending up at the reaction center, and then somehow the most efficient way is selected. Still, a number of people argue that that's not required. Quantum coherence isn't necessary, and that purely non-quantum explanations suffice. Well, so we got to 2007. 2011, Phil Ball, the science writer, wrote an article called The Dawn of Quantum Biology. It made the front page of, he wrote it in Nature, made the front page of Nature, and there's the iconic bird, the sort of the poster child of this new emerging field, because clearly there were people who were starting to think about developing experiments and coming out with experimental results that supported the idea that maybe quantum mechanics is playing a role in biology. I got interested in the subject. So as Simon mentioned in the introduction, my background is nuclear reaction theory. And in particular, I spent 2.5 decades studying the structure of light nuclei, so neutron-rich nuclei on the drip line of the Segre chart, looking at the structure and few body systems. And so, essentially quantum mechanics, but I'm trying to understand the structure of nuclei. It's around about that time, it was in the late 90s, when 97, 98, a colleague of mine at Surrey, John Joe McFadden, who's a molecular biologist, came to the physics, he's at Surrey as well, he came to our physics department to give us a talk on what it was a bit hand-wavy, the suggestion that quantum mechanics might play a role in a particular mechanism in biology called adaptive mutations. The idea being that particular bacteria, E. coli, are able to mutate in two directions, all other things being equal, A and B, with sort of 50-50 probability. But if you prepare their environment, say the water solution has lactose, which the bacteria can feed on, it changes things. The problem is the bacteria can't utilize this sugar in the solution, its environment, unless they mutate in one particular direction and not the other. And yet you see many more mutations in that direction, as though they knew in advance that that's the direction they had to go. Anyway, I think the problem, I haven't followed the literature, but I think that problem has been resolved without any call on quantum mechanics. But John J. McFadden gave this talk, and I think a lot of my colleagues were rather skeptical, to say the least, but I felt there was something in it, there's something interesting. So that's started my interest in the potential effects of quantum mechanics in biology. As Simon mentioned, John, Joe, and I then went on to write what I'm still proud to say is the only popular science book on the subject. And in the book, we go from what is more established, simple steps to the more speculative. So I think by the end, we do talk about Penrose hammer-off quantum consciousness, but by that point, we are extremely skeptical, because I certainly think that if there's something to be said about the role of quantum mechanics in living systems, we really should be walking before we can run, and take things slowly and carefully. So there are a few list of phenomena and mechanisms that we might look at to see where quantum mechanics plays a role. Enzyme action, as I'd mentioned, was confirmed by the Berkeley group in the late 80s with proton tunneling. Photosynthesis was another one that's well established, but I think the jury is still out on that as well. Magnetoreception in birds, well, it's the leading candidate, but there isn't that clear smoking gun that suggests that quantum entanglement is in fact the explanation there. Quantum tunneling is also suggested to play a role in smell. So in particular, Luca Turin's work has suggested that it might be that the vibrational frequency of scent molecules plays a role in allowing them to be recognized by the receptors in the nose, rather than the shape, the lock and key mechanism, which is the standard way we talk about how we smell. And so this vibration theory, he says, well, that might be due to the right frequency of vibrations, the energy of the scent molecule triggers electrons to quantum jump across from one site to another, and that's what triggers the signal that goes to the brain. Again, there are many people who are rather skeptical of that theory. Then there's DNA mutations, and that's the subject of what I want to talk about in the second half of this talk. There are much more speculative ideas: connections with cancer, connection with the origin of life, connection with the nature of consciousness. They are hugely speculative. And the concern is always that if you say, I'm working in quantum biology, the assumption is that you must be working in some of these areas, to the extent that many of my colleagues who are quite serious about some of this work would regard those last 3 points as woo-woo physics, not to be taken too seriously. But who knows? It doesn't, just not to be taken too seriously at the moment. Okay, we're far from being able to say anything definitive about them. The sorts of things when we talk about quantum biology, we don't mean simply quantum mechanics involved in chemical bonds. We mean non-trivial quantum phenomena. So long-lived coherence, superposition, tunneling, entanglement, and so on. Well, John, Joe, and I, having really been working on this stuff as a hobby I carried on doing my stuff in nuclear physics. John Joes was doing a lot of work on tuberculosis, and he had his own research program. And we'd meet up now and again and talk about some of these ideas in quantum biology. And in fact, we published a paper in '99 on adaptive mutations. It's been cited quite a lot. I'm not particularly proud of it because I don't think it really went anywhere. But some years ago, we decided to take it a bit more seriously, and we applied and secured funds from the Leverhulme Trust to create a doctoral training centre at Surrey to fund 20 PhD studentships. And this is our first cohort. In fact, already 2 of them, I'm stepping out of shot here to point to the audience here, 2 of them have already received their PhDs and in fact are now postdocs working in our group. We've kept hold of them. It was a really, the other picture you see here is the scribbles on the windows. I love this picture because when we first set up this open plan office for these PhD students, and bear in mind, these are physicists, chemists, and biologists. It's very interdisciplinary, and it was a really lovely, fuzzy sort of vibrant atmosphere in that office. It was a new office, newly decorated. We hadn't put the whiteboards up. So they're just reusing the windows to scribble stuff on. So I like that picture. Those windows have since been cleaned, I should say. So this was a good opportunity for someone like me who's come from a different field to be dragged along with the students in doing some more serious work rather than just over a beer or coffee with John Joe. Since then we've been able to sort of work on some aspects of quantum tunneling in biology, and I've also collaborated then with computational chemists who are able to do sort of big grown-up calculations and modeling, and that's what I want to talk about now. So what we've been working on. Remember, I mentioned Löwdeen's work suggesting that quantum tunneling might play a role in proton transfer from across a hydrogen bond from one strand of DNA to the other. So this was his paper, Proton Tunneling in DNA and its Biological Implications. When I first read this paper about 15 years ago, I think, I thought I saw it as the very first reference to quantum biology. He talks about electronic and protonic structure of biologically interesting molecules and systems has to be treated by quantum chemistry. This has led to the opening of a new field, which has been called submolecular biology or quantum biology. I wasn't aware of the work of people like Pascal Jordan going back to the early 1930s, where he also uses the phrase quantum biology. But I also mentioned that Löwdeen's paper in 1963 didn't have any sort of calculation. He has some vague diagrams in it suggesting how this mechanism might take place, but no serious calculations. But the idea is this. So the strands of DNA wrapped around each other, the rungs of the ladder holding them together are hydrogen bonds. If we zoom in, particularly looking at so genetics 101, adenine and thymine always bond together, guanine and cytosine always bond together. If A bonds to C or something else, then that's a mismatch, and that can lead to a mutation. And so you can see these nucleotides are held together by these hydrogen bonds, protons. They're basically joining an oxygen and a nitrogen atom, for example. And the idea is that, for example, between T and A, it's a double hydrogen bond. So 2 protons, and one sits closer to one nucleotide, the other sits closer to the other. There's this probability, which we need to look at, that suggests they could jump across. Whether one jumps across and the other follows, or they do so simultaneously, was something that needed to be looked at. But this jumping across, it's unequal. They're not sitting in symmetric double well potential. Each one is much prefers to stay in a particular state on a particular site. They do jump across. That takes these nucleotides to what's called their tautomeric form, which are very unstable. The question is, how unstable, how likely is it at any given moment, if you take a snapshot, that you would find these protons on the other side, and how do they get across? So one can imagine that the proton has enough energy, whether it's the environment giving it, thermal activation, giving it a kick to jump over the barrier, or it can tunnel through the barrier. Of course, it depends on what energy level that proton is sitting in and how symmetric or asymmetric this potential well is. So this is what we set out to do more carefully. So this is an animation produced by one of our PhD students, Max Winoken, that shows how, in fact, what he was looking at here is the double strand entering what's this structure called the helicase, where the two strands are unzipped. As it's entering, these protons can be jumping from one side to the other. The question was, how likely is it that those protons will be in the wrong site as those two strands separate? So you can imagine this potential well, the barrier getting wider and wider. And if you've got the protons on the wrong site, they're trapped there. You see, on the other end of this mechanism, each individual strand is then a copy is made of it. And that's in the process of replication. If that proton's in the wrong place, then A won't bind with T anymore. C won't bind with G anymore. And that could lead to a mutation. We realized that in order to make any progress in understanding quantum effects inside living systems, we would have to treat the process, the dynamics of the proton as being part of an open quantum system. Namely, action of the environment will have to play a role. But turned out there were two stages to what we had to do. First of all, we had to work out what was the potential energy surface that this proton was feeling. And that's where the chemists come in and computational chemistry work. There's a vibrant group at Surrey who use all sorts of molecular dynamics techniques and density functional theory and so on to map the potential energy surface. Once you have that, stage two is to say, right, I've got a potential energy surface. I now rewind the clock, start again, and treat this proton as a quantum system that is not isolated from its environment. It's feeling this potential, but it's also feeling the influence of the surrounding water molecules that are thermally agitating it or causing dissipation and decoherence. Well, a paper that we published last year, led by who was then a PhD student, Louis Slocum, looked at both types of the pairs of nucleotides, C and G and A and T. And you can see clearly with A and T. So what we've got here is the deep well, which is where the proton is much more happy and stable, and then the possibility that it could be transferred across to the other side. But with A and T, there's not much of a well there. It's very unlikely that that proton will survive there for very long at all, and it'll just roll back again. But with C and G, there is, on the tautomeric side, there is a dip, a well, where even though this is a very asymmetric double potential well, there is a chance that the proton could exist in there. Of course, what's happening here to generate these potential energy surfaces is non-trivial. Using something like density functional theory, one has to take into account many atoms in each of these nucleotides in the molecules, and those atoms undergo reorientation. And so with the protons jumping across, they will change their position. You have to take that into account to map an accurate potential energy surface. Once you have this, in the ideal world, you'd have the proton dynamics taking place at the same time. So it's all in one stage, one process. But we're breaking it up in two. We're having the proton go across from one side to the other to give us a static potential energy surface. Then we wind the clock, stick a proton wherever, and allow it to evolve, thinking of it as an open quantum system. Okay, yeah, absolutely.
Are those plots theoretical or empirical? Sorry.
These are theoretical plots. Yeah, absolutely. I don't know how well versed the whole audience is in quantum mechanics, but I'm sure you can cope with this. I'm keeping it very simple. If we think about an isolated quantum system, of course, we use the Schrödinger equation, but once you have an open quantum system, we go from talking about a wave function to talking about a density matrix or a density operator. The Schrödinger equation, just for an isolated system, can be recast in terms of what we call a master equation involving the density matrix. Again, first order derivative in time, and then you have the density matrix commutator with the Hamiltonian. So this is known as the von Neumann equation. There's no, this is to describe a pure state, isolated quantum system. However, what's so useful about the density matrix approach is, of course, that it allows us to describe an open quantum system, which is interacting with its surrounding environment or heat bath. There are lots of different approaches to doing that. There's the approach due to Lindblad. There are others. We start off with the model developed by Caldera and Tony Leggett back in the early 80s, very highly cited paper for people working in open quantum systems. Essentially, you have your system of interest surrounded by a heat bath of harmonic oscillators, and each of those will have a Hamiltonian associated with it, and then you have an interaction Hamiltonian between the system and bath. And that allows us to develop a more extended master equation than the one I showed on the last slide, which is just d rho by dt and then the commutator, but with additional terms. So you have the first term on the right-hand side is what's called the von Neumann term. That's just the commutator. That's unitary dynamics for an isolated quantum system. Then you have two additional terms for dissipation and thermal noise and decoherence. And to get to this model, various assumptions and approximations are made. But the basic idea is that your quantum system surrounded by an environment, essentially the environment is constantly, continuously measuring the system. The system then leaks information out into the environment. For this particular model, the Caldera-Leggett model, there are simplifying assumptions made about the nature of the environment. For example, that it's an infinite heat bath of oscillators, frequencies going all the way up to infinity. It's ohmic, which suggests that the quantity that describes the environment, the heat bath, is what's called the spectral density, is just proportional to the frequency of the oscillators. Also, because the environment is seen to be infinite in extent, then this is a Markovian system, which essentially means the environment has no memory of the system. That information leaks out and is lost in an infinite bath and never to be retrieved. So all the system knows as it evolves is the dynamics of the previous step. Well, the environment knows the dynamics of the previous step of, in the process. This model was then used to describe the proton in that double well potential, describing the hydrogen bonds between these nucleotides. Paper only came out last week before in Nature Communications Physics. Again, Louis Slocum, now a postdoc, who's taken, it's the next step to see the importance of proton tunneling. This potential is essentially, it's a curve fitted to the parameters that we get from the density functional theory calculation. So those two plots essentially give us, we have to stick walls on either side. We need to constrain the proton, can't just carry on. It's held in place and in either well. So the asymmetry delta E between the bottoms of the two wells is something that's given to us from density functional theory, as is the height of the barrier. And this is looking at a single proton between G and C nucleotides. Those lines there are just the energy eigenstates in this double well potential. But you can see it's not until sort of the sixth excited state that there is any amplitude in the shallow well. So clearly, if you had an isolated quantum system and you start the proton in the ground state, it's going nowhere. It's going to stay there. It's a stationary state. You could put it in some sort of a Boltzmann distribution of many eigenstates, but even so, there'll be a tiny, tiny probability that there'll be an amplitude in the shallow well. But stick this into an open quantum system, and what you see is the environment not only causes decoherence and dissipation, but at the same time thermally activates the proton, gives it a kick. And we found that giving it a kick up to an energy level below the top of the barrier was much more likely, obviously, than getting it right above the barrier. So if it gets across to the shallow well, it's overwhelmingly more likely to have got there by tunneling through the barrier than hopping over it. In fact, this is I just pulled a few words from the abstract of our paper. So what we find is that, so the canonical form is the standard stable form where the proton's sitting in the deep well. The tautomeric is the unstable shallow side. The canonical and tautomeric forms of GC interconvert over timescales far shorter than biological ones. That quantum tunneling happens over femtoseconds. And as far as biology is concerned, that's too quick. But that doesn't matter. What it means is at any given moment, as far as the biological system is concerned, the state of this proton is in thermal equilibrium. There'll be a certain fixed probability that you could, at any one moment, if you take a snapshot, that there'd be a fixed probability that you'd find it in the deep well or the shallow well. So it reaches thermal equilibrium rapidly, and we calculate that the occupation probability in the shallow well is 1 in 10,000, which is huge. Previous estimates were like 1 in a billion. There's a 1 in a billion chance that that proton will be sitting on the wrong on the wrong site when the strands separate, or at any given time. But we find it's much higher than that. This is the first time anyone's done an open quantum system calculation. People have used density functional theory and used various approximate chemical, chemistry techniques, or they've used WKB approximations or whatever, and they get very small probabilities. Even though we're using a Caldera-Leggett model, which is, comes with various assumptions and simplifications, like the fact that it's Markovian, the environment is ohmic, it's a high temperature approximation, which actually is fine for biology. Nevertheless, it suggests that the tunneling probability is much higher than people had thought before. The next step, something else that we're doing, a paper that, again, there was also published this year by two of our other students, Safar Lally and Nick Wareham, looks at going beyond the Caldera-Leggett model, looking at non-Markovian corrections. And again, there are many, many people working on non-Markovian corrections, and there's a whole industry here. The technique that in particular Nick Wareham developed was to say, what if the environment, so if the spectral density is just proportional to the frequency of the oscillators in the bath, what if you put up a high frequency cutoff? So you're saying, the bath oscillators don't go all the way up to infinity. There's a maximum frequency that beyond which there aren't any oscillators in the bath. Already you've gone beyond what is a Markov approximation there. And then Nick developed a technique for developing some expansion, a series expansion in powers of 1 over this maximum frequency. So this cutoff frequency is some large number. So 1 over the frequency in a power series expansion. Looking at the leading order correction, that will be the leading order non-Markovian term on top of the Markovian approximation of Caldera-Leggett. So we're putting in non-Markovian corrections. And what we see, so what you're looking at here is if people are familiar with the famous, there's a Physics Today article by Zurich on open quantum systems, where you start off with something in a superposition of two Gaussian peaks. This is a plot of the spectral of the density matrix. So the line across horizontally, those two peaks are sitting on the diagonal of the density matrix. The two lines the two bumps vertically, those are the off-diagonal elements that describe quantum coherence. The left column here, what I've called R equals 0, that's the standard Caldera-Leggett Markovian version. On the right-hand side is adding in this non-Markovian correction. And we're dropping down in time sequences. So you can see very quickly the off-diagonal elements disappear in both cases. But what we see is some weird resurgence of off-diagonal rippling effect on either side of those peaks on the diagonal, which we've called lateral coherence. There are various ways of measuring quantum coherence. We've used a quantity called the L1 norm. So it's basically a measure of just to what extent this thing has decohered and it's gone from a pure state to a mixed state. The action of the environment. You can see that the line that's at the very close to t equals 0, that's the initial rapid decoherence that we see from the first figure down to the next one. The different curves here are different amounts of non-Markovianity in our model. And what you can see is there's this resurgence of coherence.
Es kann ja noch ein bisschen schlimmer werden, aber das letzte Mal sage ich nur,
coming from someone without not turning the microphone off. Anyway, I'll carry on. It's not interrupting me. And so this is the early stages. We want to understand how important is it that our environment, we have to treat it more carefully than the Caldera-Leggett, the Markovian ohmic environment. The reason why that's important is because if in living systems, quantum coherence can be maintained for timescales that can play a role in biology, then there must be something clever going on. The environment can't simply be white noise. It can't simply be an infinite heat bath that's just sucking away coherence, like putting a hot cup in the freezer. The environment, as described by its spectral density, may have structure. There may be certain frequencies in those the bath of oscillators that are more important than others. So imagine sort of spikes on top of the spectral density. And it could be that life has had plenty of time to evolve trickery, to tweak the nature of the environment, to do with the vibrational frequencies of these molecules that act not to cause decoherence, but to somehow resonate with the quantum system itself, in this case, the proton, and maintain coherence for long enough to play a role in biology. That's still an open question. At the moment, in fact, this paper, for example, the word biology doesn't appear in it. Okay, this is just purely a theoretical physics paper. But we can, the motivation for doing it is that maybe there's something happening in biological systems that Erwin Schrödinger would have been very pleased to hear about, that maybe we have to look more carefully for. I want to end just by saying that I set up a virtual quantum foundation center at Surrey to bring together various people who are interested in some of these ideas. And also because, as a nuclear physicist and in the tradition of my PhD supervisors, I sort of was discouraged. I was talking with Simon about this earlier, discouraged from talking about foundational issues in quantum mechanics. If you weren't a Copenhagenist when I was a student in the 80s, it's sort of, you had to keep your head down, right? You whisper, I quite like Bohmian mechanics, and even weirder if you're a many worlds advocate. And so I've sort of reached that stage in my career where I'm quite happy to sort of explore some of these. And also, I think we're getting to that point where a lot of physicists feel we have more freedom, to be more open about some of these foundational issues. So I'm sort of linking that in with some of these ideas in quantum biology. Recently, we received a large grant from the John Templeton Foundation called Life on the Edge, Quantum Thermodynamic, Quantum Biology, and Arrow of Time. It's basically a collaboration of 6 universities led by Surrey, including Oxford. So at Oxford, of course, Simon is one of 3 philosophers of physics who are named on the grant that we're working with. We also have Vlatko Vedral and Chiara Marletto here at Oxford, who are part of the team. Then we have other philosophers, Karim Thibault at Bristol, Eddie Chen at San Diego. We have Paul Davis at Arizona, and we have a group of experimental quantum biologists at UCLA who are carrying out some experiments. There are lots of different strands to the work. So we're looking at open quantum systems and biology. The big motivation for this project is really on issues on the arrow of time. And, while listen to someone like, Sean Carroll will tell you that everything in the universe is time reversal invariant plus the past hypothesis. So, so the arrow of time is only about increasing entropy. You don't need to worry anywhere else. I'd like to see if I can understand more about the irreversibility of the arrow of time down at a quantum scale when we go from an isolated to an open system, linking with things like the measurement problem. And that's where it links in with foundational questions in quantum mechanics. And so this is a really exciting group. We have 16 people named in the grant, 3 philosophers, half a dozen physicists. We've got a couple of computational chemists. We've got 2 applied mathematicians and 2 molecular biologists. And we'll see where it goes. I just wanted to then end by acknowledging both Leverhulme Trust, of course, for our Doctor Training Center and the John Templeton Foundation for funding this new project. I've mostly worked with John Joe McFadden, molecular biologist. Andrea Rocco is a fellow theoretical physicist. He comes from statistical mechanics. Marco Sacchi is a computational chemist. Then we have our PhD students. And I want to leave you with perfect timing. It's very good, isn't it? I want to leave you with an announcement of a forthcoming meeting. Part of our John Templeton Foundation project is quite a large activity in outreach and doing lots of different other fun things. Somewhere between our head down work and sort of sticking our head over the parapet are what we're calling a series of blue sky thinking workshops. So they're very similar if you're familiar with the FQXI series of meetings where people are deliberately provocative and speculative. And so we have the first one happening this July. It's entirely online. Next year, we're hoping it'll be face-to-face at UCLA. And then the following year, we'll be back in the UK, somewhere in London. We already have a program. This is going to be announced. So in fact, I'll be very happy if you have a mailing list that we can send this announcement round. So we have our speakers already. So it's a mixture of physicists and philosophers. The idea is that these talks, that people are free to say what they want to talk about, and as long as it's loosely connected with some issues to do with the arrow of time and irreversibility. And so we hope as many people as possible can join us online. But otherwise, thank you all very much for your attention.